Chapter 27 is an introduction to animal diversity. It takes a look at the features of the animal kingdom and then how we classify animals. So to be an animal in general, you have to show multicellularity, so you're a multicellular animal. Most have complex bodies, all but Porifera do, essentially, with complex tissue structure. All animals are heterotrophs. We have to ingest and digest other organisms. We all show active movement at some point in our life. There are some that are sessile as adults. There's a lot of diversity in form and size, so you can have microscopic animals to animals that are absolutely enormous, so there's no kind of finalized sizing. Most of us show sexual reproduction. There are some asexual examples we'll look at. Offspring pass through developmental stages. They can be as complex as metamorphosis in some arthropods or human development where you have a defined, baby stage, adolescent stage, and adult stage. Animals do have fixed body plans that are determined by genetic cues. Hox genes control what we're going to look like. Essentially what that means is you can have all the characteristics of an oak tree. I can know for sure that you're an oak tree, but it doesn't necessarily mean that I know how many leaves you're going to have or what your branching pattern is going to be. When it comes to an animal, however, I know how many limbs you should have and where those limbs should be located. That's a reference to that fixed or determined body plan. For the complex tissue structure of animals, we all lack cell walls for support. We don't need them. Our bodies make up that kind of support need in a different way. We also have unique intracellular communication mechanisms called gap junctions. We have 4 different kinds of junctions. You've probably studied them in cellular biology. In animals, you have plasmodesmata, you have those gaps for cytoplasmic streaming in fungi, but unique to animals specifically are gap junctions. Our tissues can be divided into 4 categories. Connective tissues are any cells that are embedded in an ECM, in an extracellular matrix. So bone, cartilage, blood, those are all connective tissues. Your epithelial tissue, that's any tissue that lines or covers, protects, or secretes. So if I line the inside of an organ, I line the inside of your mouth, I protect the outside of your body, or I'm secreting something like sweat or a hormone product, those cells are epithelial cells, and together they make up epithelial tissue. Nervous tissues coordinate movement. Those are your nerves in your brain. And then muscle tissues, of course, power locomotive activities. So you can look at muscle tissue can be divided into smooth, skeletal, and cardiac muscle. The study of those tissue types is histology. It's its own branch of biology. Animal reproduction and development, as I mentioned previously, most animals undergo sexual reproduction, and they're diploid organisms. The majority of cells in their bodies are diploids, and then their gametes tend to be haploid. The majority of animals have haploid egg and sperm that unite through some sort of fertilization process. You end up with a diploid zygote that then grows, via mitosis. That's quite different than fungi that have 2 different mating types, and then protists and prokaryotes that have incredibly variable ways that they can go about reproducing. So there are some exceptions to that to that sexual reproduction plan. There are just animals that have an asexual phase of their life cycle. They tend to be smaller invertebrates or more simplistic arthropods, hexapods, specifically insect males. There are a few of them that are haploid. Some animals grow via budding or fragmentation, so they have essentially a small part of their body that breaks off and becomes free living. Hydra are an excellent example of that. If you look up hydra budding on YouTube, there's a lot of really fantastic videos. And sea anemones can do the same thing. Some animals grow via parthenogenesis, which I've always found fascinating. In parthenogenesis, unfertilized eggs can grow via mitosis into male individuals. It's a pretty cool mechanism. There are some vertebrates that do this, like chickens, and then there are quite a few insects that are capable of doing this. The problem with parthenogenesis is that because you don't have 2 parents worth of genetic material, you can really build up those mutations. A lot of times genetic mutations can be masked by whatever genetic information you got from your other parent. But if you only have 1 parent, you obviously can't mask whatever that defect is. When it comes to overall reproduction and development, animals tend to follow this generalized plan. So we said that a male and female gamete come together to form a zygote, which grows via mitosis into a solid 8-cell ball stage after 3 divisions. Usually, if you're going to have twinning in an animal, that's when twinning happens. That 8-cell ball will break apart. Otherwise, it starts to grow via cleavage, more cellular divisions, into a hollow ball of cells known as a blastula. You can see the blastula in cross-section right there. And it's got this blastocele, this specialized internal cavity. And that internal cavity can develop into a few different things, as we'll see in just a moment. When you evaginate that blastula, you end up with gastrulation is the process. You get a gastrula. So that gastrula has an opening, a pore-like opening, and then essentially what becomes a gut chamber, and you can end up with either 2 or 3 germ layers, and those germ layers will develop into different tissues depending on the animal that you're studying. It gets super specific, and we'll look more closely at that in later chapters. So we're just kind of keep it basic for right now and add a little bit more information as we go. All of this development, as I mentioned previously, is controlled by homeobox or Hox genes. These are master regulatory genes. So what they are coding for are those variety of transcription factors that dictate when other genes and gene clusters get turned on and off. It says, where we're going to place appendages, how many appendages we're going to place, what those appendages are going to look like. So they don't necessarily code for all of the tiny things of, what makes up an arm, but it does say, hey, you should put an arm, let's have 2 of those. They're going to be bilaterally symmetrical towards the head end of this animal coming out of the shoulders. That's what a Hox gene is going to control. So when we have more Hox genes, what you end up with are more animals that are a little bit more complex, that have a little bit more organization to their bodies. We see that Hox genes have just been repeated over time, so we're duplicating Hox genes, and that's leading to more complex organisms as we study our animal phylum. We use these different features to classify animals to create this large phylogeny. We look at things like, symmetry, and we're going to touch on all these things in just a moment if you don't understand what I'm saying, but we look at things like symmetry, the number of tissue layers that you have, the origin of the mouth and the anus. Was that original blastopore? Did that become your mouth, the entrance to your digestive system, or did it become the exit? We look at your body plan and the number of body cavities you have and within those cavities in order to classify you. All of the individuals that you see in this little this portion of the phylogenetic tree of kingdom Animalia, all of these guys are invertebrates, except for that little chordata. So if you break off the deuterostoma, you have the chordata. Members in that little group, they all radiate out and become our invertebrate individuals. So you can see it a little bit bigger here, all of these characteristics that we use to classify animals. The first one I mentioned was symmetry. That really is the easiest way to start splitting off animals, because we look at if you have symmetry. If you do not have symmetry, if you're asymmetrical, then that automatically puts you in phylum Porifera, and those are the sponges. The sponges are the most basic of animals, and we'll look at them really closely in the invertebrate chapter. They just kind of grow essentially in whichever which way they can grow. Sponges lack, technically, if you want to get super technical about it, they lack true tissues. One of the creepiest experiments I've ever seen as an undergrad, or creepiest demonstrations I've ever seen as an undergrad, you can put a sponge in a blender, make a little sponge smoothie, and if you pour it back into healthy seawater, pour it back into that healthy water that you got it from, it will reform into a new sponge. It's quite strange. So that's Porifera. They're not terribly complex, and they don't particularly mind how their cells are arranged. The second type of symmetry is radial symmetry. So in radial symmetry, that's what you see in the middle picture. So your body is arranged around a central axis. You radiate out. Think radial tires. You're essentially organized into a circle. So rather than having a clear left and right, you have an oral and aboral side. So you have a side that has a mouth and a side that has a back, essentially. So this works really well if you are either stationary or you just float through the water, because they don't have to be super directional. Things that are circular in nature don't tend to swim often in any particular direction. They can kind of go anywhere. So it makes sense that they live in the water. They don't have to travel along a straight line. Cnidarians are the most famous example of these. Jellyfish are radial in symmetry. Things like sea anemones are radial in symmetry, and you'll get to see more of those later as well. The most complex body plan when it comes to organization is bilateral symmetry. So that means that you're divided along the sagittal plane. The sagittal plane is the plane in anatomy that cuts you into left and right, and you have a clearly defined left and right. Half of you matches the other half of you, and then you have a head end. That's cephalization. The process of cephalization is having a head end and a butt end. Cephalization allows for the collection of sense organs in the head. So if you think about it, your 5 senses, sight, hearing, smell, taste, and touch. You can touch, obviously, with all of the organs or all of your skin, but the rest of those organs are centrally located in your head, and that gives you a really great kind of sense of direction for moving forward in a straight line. All other animals outside of members of Porifera and then those cnidarians and the ctenophores, everyone else is bilaterally symmetrical. There are some animals, you'll see them in adults and you're like, wait a minute, that thing is not bilateral. They are called bilaterally symmetrical because they were bilaterally symmetrical as larva. One of my favorite animals is called a tunicate. They're really cool looking, but they look very similar to a sponge. If you're not quite sure what you're looking at, they look like a translucent sponge. So they definitely don't appear to have bilateral symmetry, but they do when you study them as larvae, and they just lose some of that specialization as they enter into adulthood. Another way that we classify animals is on their embryological development, looking at the features of them as embryos. We can divide them into 2 major groups, the parazoas, the beside animals, as previously mentioned. Parazoa, that's phylum Porifera. They don't have any true tissues, hence why we can put them in a blender, and they don't show symmetry. Everybody else are the eumetazoas. These have distinct tissues and a distinct symmetrical plan, be it radial or bilateral. When we study animals, we've mentioned this before, you can be a radial animal and you are a diploblast. That means you have 2 germ layers. You have an ectoderm and an endoderm. You have an outside and an inside. Those are those 2 phylum previously mentioned, or you can be bilateral. If you're bilateral, you have the option to be a triploblast. So that means you have an ectoderm, an outside, a mesoderm, a middle, and then an endoderm, an inside. Ectoderm and endoderm, your outside and your inside are kind of obvious. The outside is your skin, the inside is the digestive tract, your alimentary canal, everything from mouth to anus. And then mesoderm, those are the meaty bits of you in between. All of your visceral organs are in your mesoderm. The vast majority of animals that you are probably familiar with are bilateral triploblasts. Again, for those different layers, for your endoderm, what that develops to eventually is the lining of your digestive tract, your alimentary canal that I mentioned previously, your trachea, and your lungs. The mesoderm's the stuff in the middle, muscle, bone, cartilage, blood, and your organs. And then the ectoderm are the things on the outside of your body, and actually your CNS, your central nervous system. So there's a couple different ones you can throw in there that aren't quite as obvious. So again, for that embryological development, you can look at the diploblasts or the triploblasts having defined body cavities. It's actually what distinguishes those triploblasts. So having a defined body cavity, that's what grows out of that mesoderm really, really, really early in development, and you have a body cavity, a space between your body wall and your visceral organs, and it tends to be fluid filled, especially in humans if you're focusing on human biology. And that's actually really helpful, the fact that it's fluid filled. It helps with shock absorption, but it can cause some interesting problems. If you've ever heard about someone having fluid around their heart, it's that fluid that lies between the body wall and the visceral organs, in this case trapped under a pericardium, that can cause some issues. In animals that aren't as complex as us, it can actually, that fluid that they hold can create their hydrostatic skeleton, something you'll learn about in phylum Echinodermata. starfish. Starfish are really great at using that fluid to move around. You can put organs within your coelom, as we've mentioned before. Our organisms, when it comes to organizing them by presence or absence of their coelom, they're acoelomates, so they don't have one. Pseudocoelomates, they have something that kind of looks like a body cavity, but not really. And then coelomates, those are all the ones that do have a defined cavity with a defined purpose. Let's look more closely at those. So for acoelomates, they have a defined body cavity. The mesoderm, rather than being any sort of defined organ, it's just kind of chunky tissue. Phylum Platyhelminthes is how you say that fun word, the flatworms. They're excellent examples of acoelomates. So if you touch them, they feel like very firm animals, but they don't have defined visceral organs. They're just filled with tissue. So they're acoelomates. Your pseudocoelomates have a false body cavity. So that body cavity, the tissues, the things in it, are growing from the endoderm and the mesoderm in. So from the endoderm in, essentially out towards the middle, and from the ectoderm in, if that makes sense. It's not mesoderm derived. It can have a function. It can host that hydrostatic skeleton that we mentioned before. It can help them to move. It can help them be more firm. It can help them to withstand kind of harsher environments. Nematodes, roundworms, tend to be parasitic, and one of the reasons that they do so well in really harsh internal environments inside of other animals is because they are pseudocoelomates. They're very firm creatures. Last but not least are our coelomates, or true coelomates. So in this case, that body cavity is derived entirely from the mesoderm. It's body cavity space with tissues for shock absorption, or fluid for shock absorption, and then all of your internal organs. The tissues that are mixed in there in the body cavities with your internal organs, it holds everything in place, and it allows for motion, because if all of our organs were kind of stuck together, you would have a really rigid abdomen. All animals would have really rigid abdomens, and it would impact the ability for our bodies to be flexible, which is one of the really great things that comes from our spines, actually. If you can imagine having a spinal fusion, if you've ever seen someone that has that surgery, they have a really hard time moving around, and that's what would happen if you didn't have those tissues that are separated by these different cavities. You'd be really, really stiff. So again, presence of absence of the coelom makes you an acoelomate, a pseudocoelomate, or a coelomatic animal. We can further organize our triploblasts. We can kind of further differentiate them by their embryological development. We can look at the formation of the mouth and the anus, how their cells cleave, if they cleave in straight lines or radial patterns. We'll look at it in just a second, and we'll look at how that coelom forms. So again, we're just kind of breaking down even more, how do we characterize animals as we have all these branches that split off as our animals get more and more complex. So the formation of the mouth and the anus, that blastopore that we mentioned before, so that first opening that happens in the formation of the gastrula stage, you're called a protostome. That original opening becomes your mouth, and you are a deuterostome if the original opening becomes the anus. Both of these things end up with a second opening, but it's just kind of what develops first. You fit in that deuterostome group. I remember feeling quite smart when I told my older brother that at one point in time in his life he was nothing but a butt, and he never grew out of it. I thought that was the coolest science joke in the world when I was younger, because I was a huge nerd. But that's one way that you can further classify your triploblasts. You can look at cleavage furrows, or cleavage patterns too, excuse me. So for protostomes, so those organisms where the very first opening, the blastopore becomes a mouth, they can have spiral cleavage, so their cells literally are splitting from one another in a spiral pattern along the axis in the embryo, if you drew a straight line through the embryo, or they can have determinate cleavage. So that means that every cell in that organism knows exactly what it's going to be very, 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 very early, when that spiral cleavage is a little bit more flexibility. For deuterostomes, we undergo radial cleavage, so cellular division at very specific right angles. In some cases you have indeterminate cleavage. So that means that you have the fates of some of your cells are determined a little bit later in development. So this again leads to slightly more flexibility, slightly more diversity in the animal species that you see or what individuals of that species can look like. For animal phylogenies, so all of these characteristics that we've been talking about help us to organize animals, to look at kind of what groups they belong in, define phylum. For a lot of our history as biologists, we were classifying animals by their morphology, by what they look like. And that can be super misleading. Anyone who studied evolution knows that. Just because you have wings does not mean that you're closely related. And we also know that sometimes, certain characteristics are lost. I mentioned those tunicates before that I really like. If you're only ever studying adult tunicates, you're going to think that they're really closely related to porifera, to sponges, because that's what they look like, and couldn't be farther from the truth. So it's really hard to study phylogenies from just a morphological basis. Modern systematics or systematists, the scientists that are trying to create more accurate phylogenies, are using biochemical, molecular, and genetic evidence to build these phylogenies instead of just what you look like. That has made our phylogenetic trees a lot more reliable. Again, we still have some of those original definitions. We got it right the first time. We know that all eumetazoa with differentiated tissues are more closely related to each other than they are to the parazoa, the animals that lack tissues. We have found through molecular evidence that we think that both of these groups even still evolved from a common ancestor that looks super similar to a modern day tranoflagellate. So those cells are protists. So that original organism is a protist that has a flagella on it that allows it to push itself backwards rather than propel itself forward. I realize it sounds a little funny, but the flagella is essentially on the opposite end of what you would think. And we're relatively certain that animals evolved from a common ancestor from this particular protist group. So from there, the eumetazoa are divided into two major clades based on their embryology. So do you show radial symmetry, and that splits that eumetazoa group into the cnidarian clade and the ctenophoric clade. So into the stinging animals, they have those cnidocyte cells and then comb jellies. And you'll learn more about the specifics of all of these groups in the invertebrate chapter and the vertebrate chapter. So if you feel like I'm just spitting a bunch of information at you, it gets better, I promise. So eumetazoa, we can divide them into two clades, those with radial symmetry and those with bilateral symmetry. So thus far, we've actually only isolated three groups of animals. We've isolated our porifera, our sponges. We've isolated our stinging animals. We've isolated our comb jellies. Everybody else fits into this bilateral group. The bilateral group is then split again. We split it into two more clades, again based on embryology. We split it into our deuterostomes. Deuterostomes are the animals, remember, whose blastopore becomes the anus. These include echinoderms, phylum Echinodermata. Those are our starfish, brittle stars, things of that nature. And then chordates, chordata, that's you. You fit in that group. Chordates are animals that share four major characteristics that, again, you'll study next chapter. The other big bilateral division are the protostomes. Protostomes develop their mouth first, and you can be a Lophotrochozoa, say that 5 times fast. That's a reference to the structure of your larvae or having a specific feeding structure, or you can be an ectozoa. Those individuals molt their exoskeleton. So that includes your arthropods and your nematodes. Arthropods are centipedes, millipedes, hexapods. Oh, who am I forgetting? Your spiders are arthropods, and then all of your crustaceans are arthropods. And nematodes, as previously mentioned, are roundworms. Our modern understanding of phylogenetics, and again, the study of all those organisms is going to come in the next 2 chapters, has really developed over time, and we're still figuring out the best way to organize these animals. So I don't stress that students memorize phylogenetic trees. because they're always changing. The big organizations have stayed the same for a while now, but all those little things are really changing. We're starting to focus on mitochondrial and nuclear DNA, and then ribosomal sequences in order to put you in the appropriate group, because those seem to be the things that are most consistent over time. So molecular technology is going to continue to contribute to this study of evolutionary relationships. It's going to continue to make our phylogenetic trees more robust and more reliable. On kind of the backside of studying these phylogenetic trees and studying animal diversity, what we can end up looking at is where animals came from and kind of how they've changed over time. So what we found, or what we think, is that in the Precambrian time frame, about 635 to 543 million years ago, is when we had that original choanoflagellate cell that developed into the first sponge, that evolved into that first sponge-like organism. They have very similar DNA sequences, and that's when we first think animals had evolved. That's been pushed back. I was taught when I was obtaining my bachelor's degree, I always learned about 525 million years ago. So now they're pushing a little bit back as we get more evidence. The Cambrian period is when we saw the most rapid expansion of animal evolution. We had a lot more, a really great expansion of phylum. Most of today's phylum would have evolved during what scientists refer to as the Cambrian period. So those 542 to 488 million years ago, you saw a big explosion, is what they refer to it as, because we went from having basically one animal type to having thousands. We think it might have had something to do with rising O2 levels. Increases in oxygen would have come from actually bacteria and other organisms developing photosynthesis. Byproduct of photosynthesis is, of course, gaseous oxygen. So you would have had, oh, excuse me, that's my dog. You would have had rising levels of O2 and then rising level of calcium levels as organisms died and their bodies compacted in the ocean. So we think that would have allowed for greater mitochondrial activity, greater metabolic activity in animals, which would allow for faster changes as they survived longer and were capable of doing more. But we're not really sure. We know that there are a lot of shallow seas, so you would have had a lot of variety in ecologies, and that could have pushed quick evolutionary changes. You had a lot of changes to predator-prey relationships. There might have been a lot of genetic innovation, so maybe animals saw accidental duplications of those Hox genes. So you got a lot of increased capacity for complexity right away. There's all sorts of things that might have happened. We're probably never really going to know, but this is a really interesting area of scientific research, trying to understand what it was that happened during that Cambrian explosion. Post-Cambrian explosion, there were some pretty dramatic climate change events that happened much more slowly than our current climate change event. But what we see with these really drastic shifts in the environment of the biosphere are really major losses of diversity. The Permian and Triassic boundary would have been the greatest extinction event ever seen in the history of the world. We're not really sure if it was due to extensive climate change caused by volcanic activity or something else, but we know that there's a huge shift in the livable conditions, and you saw a giant decrease in the number of species that survived. One of the things about these mass extinction events, though, is that after the event and the environment has the ability to stabilize, you end up with really great options for adaptive radiation. So because of this Permian-Triassic boundary, this great dying, you end up with a relatively small amount of animals that now had almost no competition. And they evolved really, really, really quickly. And these trilobites, they kind of look like bugs. They look like roaches, honestly. These trilobites and these reptile groups would have radiated really quickly and become all the dinosaurs that we all studied and loved when we were 6, 7, and 8 years old. And then some of those protists those green algae looking things would have been able to move on land because of the changes in the environment. We have studied the CP boundary, the Cretaceous and the Paleogene. The Tertiary boundary is another really big mass extinction event. This one, we're pretty sure what caused it. There's a really large meteor that hit the Yucatan Peninsula, and it kicked up so much dust. It basically blanketed the planet in dust. And if you have dust on all of your plants can't photosynthesize, and you run out of all of your food sources. So you would have had really severe climate change, and you would have had a lot of dying off of herbivores and carnivores. So only really small animals would have been able to survive. And at that time frame, the really small animals that were around were shrew-like mammals and birds, and then some of the smaller plants. So those individuals were therefore able to undergo adaptive radiation, and we see the beginnings of the world that we have right now. There have been 5 mass extinction events that we currently know. There are quite a few scientists who believe that we are currently in a 6th mass extinction event, and it seems to be human-related or human-caused. It depends on the terminology that makes you most personally comfortable. To be honest, there's a really interesting documentary that OpenStax has pulled out for you. You can see the link right here. It's easy to find, on a potential 6th mass extinction, and it's really, really interesting, and it's where I suggest you go next.